Bismillahirrahmanirrahim. Hello, guys. Hope that you're good. Today, I'm going to talk about two new immunological therapies that are in the development for the cure of hepatitis B virus infection. Uh, there are only three slides that I would show you, and I would tell you about the therapies that are some of the preparations are already there on the market, and these preparations are being tried against hepatitis B, and they have very encouraging results. uh, One of the reasons that why the treatment regimes, different treatment regimes that people are trying against hepatitis B virus infections are not very efficient to treat the condition or to cause the complete removal of the virus from the system of an individual is the exhaustion of the T cells, specific T cells which target the cells which are infected with hepatitis B virus. In our bodies, there are specific T lymphocytes or some white blood cells which are known as the CD8 positive T cells, and these T cells actually or virus specific T cells or the hepatitis B virus specific T cells. They kill all the cells that are infected with the hepatitis B virus. For instance, this is a liver cell, and if it is infected with this hepatitis B virus, this cell is going to detect this particular infected cell and kill it. Now, what happens during the chronic infections that when the infection, hepatitis B virus infection is prolonged. In that case, this CD8 positive T cells or this lymphocyte is exhausted during the course of this disease, because the disease has been prolonged and the virus, these T lymphocytes are engaged in so many different types of, functions. There are other cells that also impact the working of this particular cells. For instance, during the chronic infections, these CD4 positive T cells also go down, their number goes down as well, and these cells are helping out the T lymphocytes, the CD8 positive T lymphocytes, but when their number goes down, then that help is also not coming in order to give some space to the CD8 positive T cells, because these have to make specific, these are going to help with making of specific antibodies for the same virus. And these cells are going to directly kill these cells. So for instance, in the system, people have seen that in chronic infection also these T regulatory cells, this is another type of T cells, these also are secreting lots of different substances. Similarly, the dendritic cells in our body, they are also secreting different types of substances, just like this substance interleukin 10 and this TGF beta, transforming growth factor beta. And these are directly causing the exhaustion of these CD8 positive T cells. So when you are going to find virus specific CD8 positive T cells in chronic infections, you would always find that these cells have gone down. And when their number goes down, then these infected cells, which already have the viruses inside them, these cells are not going to kill them because there are very less number of these cells in the system. So the new therapies that I'm going to talk about are, um, uh, going to, increase the number of these CD8 positive T cells, because when these cells, the number of these cells is increased in an individual who is infected, then these infected cells of your body can be killed by these cells, and the virus can be, this condition can be cured. Solution to this problem, that the T cell exhaustion, that the cell is exhausted and now there's something should be done to increase their number and efficacy. or to make them more efficient against the virally infected cell is the new immunity boosting therapies that I would just show you in the next slide. So this is quite a new study which was published on 14 May 2021. This is the latest study in this study, which was published in a Nature group of journals, they are targeting the human acyl CoA. This is an enzyme, cholesterol acyltransferase, as a dual viral and T cell metabolic checkpoint. This is a very technical, heading, so many people would not understand it. But this particular enzyme, which is given here, ACATA, this enzyme actually is responsible for, generation of these ketone bodies, or the degradation of the ketone bodies, or you can say accumulation of the cholesterol inside your body. So inhibitors are substances that inhibits that particular enzymes were tried in this particular study, and the inhibitor which was used is Avasimibe. Avasimibe, it controls the cholesterol levels, and it was synthesized, manufactured by Pfizer Group, but later on in 2003 they stopped, synthesizing or making this particular product. But still, in research studies, people, are using this particular drug in order to see if this drug has got any efficacy against other conditions. So in this group, Natalia et al., they use this particular drug, Avasimibe, in order to, see the effect of Avasimibe against the hepatitis B virus infection. And they found out that this drug is not only, protect not only has antiviral activity against hepatitis B virus, that the virus replication somehow inside the cell is being checked or kept under control by this the use of this particular anti cholesterol drug, but it also at the same time boosts protective anti-HBV T cells and anti-hepatocellular carcinoma T cells. So these are the T cells are specific to different conditions. They are specific for hepatocellular carcinoma. When the carcinoma develops, there are specific T cells which targets the carcinoma cells, and there are specific T cells that are targeting the HBV infected hepatocytes of the liver cells. So they saw that by using Avasimibe, it had this dual effect. On one hand, it decreased the replication of the hepatitis B virus inside the liver of individuals who were infected, and on the other hand, this particular drug also increased the number of these CD8 positive anti-hepatitis B virus T cells, or the T lymphocytes in the body. So they demonstrated that by using this particular drug in cell cultures, it has got very promising results, and this probably would go into the clinical trials. And if the T cell exhaustion problem is solved by the use of Avasimibe, then nucleoside analogs along with Avasimibe would lead to the complete cure of the hepatitis B virus infection, because the immunity will be boosted in that case. Another study which reported recently in 2021, again, this study was also published in 2021. They reported the restoration of HPV-specific CD8 positive T cell responses by sequential low-dose interleukin-2 treatment in non-responder patients after interferon alpha therapy. And this study reported that interleukin-2 therapy significantly increased the frequency and function of HBV specific CD8 positive T cells, which translated into improved clinical outcomes, including HBeAg, the envelope antigen seroconversion, among the non-responders chronic hepatitis B patients. Our findings suggest that sequential interleukin-2 therapy shows efficacy in rescuing immune function in non-responder patients with refractory chronic hepatitis B. So patients who this study in 2021 reported that if you use interleukin-2 along with other nucleoside analogs that people are using, or along with interferon therapy against hepatitis B virus that people are taking, if you also take interleukin-2 along with that, it would also boost up your immune responses against the hepatitis B virus. And they saw that it is in chronic hepatitis B virus patients, it was very effective. And even among the non-responders who did not respond to any other therapy previously were also treated by the use of interleukin-2. And this particular product is on the market since very long because it is used in different types of carcinomas like renal carcinoma and melanoma. This particular interleukin-2, this drug is used, and this is the trade name aldesleukin, which is on the market and it is being used for different conditions. So they use the same product for in the case of chronic hepatitis B virus patients, and they found that it is very important that it is very effective in restoring the function of these CD8 positive T cells. And when the function of these CD8 positive T cell is restored, as I showed you in the very first slide, these viruses, these lymphocytes, when their function is restored and their number is restored, these cells, are very active to kill these virally infected cells and to get rid, of hepatitis B virus once and for all. So these are the new immunological therapies, very promising therapies. Clinical trials are being conducted. Uh, but as I told you that these products already are on the market, and uh, you know, uh, I hope so. Uh, if you look at the tolerability and other, uh, issues related to different drug types, it would, take far less time for uh, these drugs to be approved for the hepatitis B virus infection as well. So that's the good news. Thank you very much for watching this.